Okay, welcome, thank you. I'm delighted to see a Friday night turnout at an alternative lecture series day. And I'm also happy to introduce Dr. Sonia Batten, who is originally from England, I believe, but lives in Nanaimo, British Columbia. And Sonia and I and many other folks, about a group of 40 total, are involved in Gulf Watch Alaska, which is the long-term monitoring program of the Exxon Valdez Oil Spill Trustee Council. And she's a biological oceanographer. She works on a long-standing project. She looks at plankton and ocean conditions in a very interesting way. And we just have had a few days of our annual meeting in Anchorage, and she was very kind to make a very inconvenient detour through Cordova on her way home to Nanaimo, which we really appreciate. When we have scientists come from so far away, we love to capture their expertise and share it with you all. So I'm really glad that you decided to show up, because she's got some really interesting things to tell you. Thanks, Sonia.
Well, actually, although it's not exactly on my way home to come through Cordova, it's been really nice to have a day here. It's a beautiful spot that you have here. It's been lovely to see it, especially on a day which isn't raining. The sound is lovely. So, I'm not very good at thinking up titles, and this was the best I could do when I was asked for a title. But what I'm going to talk to you about today is how we use some of the biggest things on the ocean, big ships, to give us an understanding of what's going on in the littlest stuff in the ocean, the plankton. And as Katrina said, I'm part of the Gulf Watch Alaska group. I get my funding for this project from a variety of sources, of which Gulf Watch Alaska is one. And I'll thank people again at the end, but I'm very grateful for the funding and contributions we do receive. So I know there are some people in the audience who know a great deal about plankton, but for those who don't, I'll start off with the basics. So plankton basically are the multicellular, the single-cell, very tiny, microscopic plants and animals that live in the upper part of the ocean. And they're the basis of the marine food chain. They support all of the marine food chains that we have. The word plankton comes from the Greek word for to wander, called planktos. And basically, plankton have very little control over where they go in the ocean. They can move around a little bit, but basically they're at the mercy of the ocean currents. And this is just a quick sort of example food chain. You start off with plant plankton here, which, just like plants on land, takes energy from the sun. They grow and reproduce. They feed the smaller animal plankton. This is krill, which most people have heard of. These are copepods. And then these feed smaller fish like herring, sometimes directly the largest animals, sometimes directly whales, but seabirds and sharks. So in this way, the energy from the sun gets passed through the food chain up to the large animals that we think of as the important resources for us that we take from the sea. There are lots of different types of plankton. There are almost as many types of plankton as there are types of animal and plant on the earth. So I just got a few pictures here of some of them. Grouped down here are the plant phytoplankton. These are usually single-celled. They're kind of green. They have the green pigment in them that absorbs the sunlight. Some of them form chains, so they can get up to that long. But they're all single-celled. And they have to stay up in the surface waters where the sun's light can give them the energy that they need. And then we have two types of animal, or zooplankton. This group here are those that they just spend a little bit of their life cycle in the plankton. So I've got here a picture of a crab larvae. This is a herring larvae. This is the larvae of a sea urchin or a starfish, and this is a little octopus larvae. So they're up in the plankton pretty much as a distribution mechanism. This gets them away from the place where their parents were. Clams also have a planktonic larval stage. So they move around, and they can find new places to settle out, new places to colonize. So they spend a little bit of time, maybe a few days, maybe a few weeks. And then we have a group of animal plankton over here that spend all of their life cycle as a member of the plankton. And they're a very diverse group. So we have single-celled animals like foraminifera that have a hard calcium shell on them. This is a multi-segmented worm that spends all its time basically swimming in the surface. This is a predatory arrow worm that actually eats other plankton. These little things are called copepods. They are the most abundant multicellular animal on the planet. So if every person on the planet had a share of the world's copepods, we'd have 1 billion each. roughly. So they are abundant. You can think of them as kind of the bugs of the insects of the ocean world. This app down here is a marine snail. It has a sort of triangular shell inside it and kind of butterfly wings on the side, and it kind of drifts around like this. This is one of the groups that's particularly vulnerable to ocean acidification because its shell will start to dissolve as the oceans get too acidic. And then the krill is probably one of the biggest types of plankton. Some people consider jellyfish plankton too because they drift around a lot, but otherwise krill's one of the biggest members, and that gets up to a couple of inches long or so. So a really diverse mixture, and they're all around together in the upper surface. Why should we study plankton? Why should we look at this stuff? Well, like I said, all marine life depends on it one way or another. Either they eat it directly, or they eat the fish that are fed on, the smaller fish that are fed on the plankton. So at some point, everything comes back to plankton, and it's important for all marine life. Because they don't swim very much, they're basically at the mercy of the currents, and they're exposed to whatever conditions are present there. They can't really get away from them. Unlike birds or fish or whales that can swim away from an unfavourable place, plankton can't, they stay there. So changes in their numbers might tell you something about how good that place is. They reproduce very quickly, especially when compared to some of the larger animals in the marine world. So typically a few days, a few weeks, the longest a year. So if things are either favourable or unfavourable, their numbers will change quite quickly, giving us a chance to measure that quite quickly too. They're not really fished. There are a few places in the world that are harvesting krill and making some kind of health thing based on that. So, but they're not fished really in an economic way yet. So we don't have to factor in fishing effects when we're looking at their numbers. So for all these reasons, it makes them good ocean indicators, or tells us something about the health of the marine ecosystem. I borrowed this image from a colleague who works down in Antarctica in the Southern Ocean. He calls this the truth slide. So the blue dots are scaled to represent the weight of that particular group. So this is the phytoplankton. These things here are other planktonic organisms. And then in this little bit here, represents 2% of the weight of living life in the Southern Ocean. This is the fish, squid, seals, whales, and the penguin dot you can't even see. So these are the things that we see as important resources, but they're supported by this vast amount of life that we don't really see. So it's an important part of the ecosystem that we need to keep track of. And again, to show the importance, this is an old slide, going back decades actually, this was put together. And this is the sort of example of the herring food chain. So in green down here we have different types of plant plankton, the pinky colour are different types of animal plankton, and then the blue is the herring at different stages in its life cycle. So when it's really young and small, it's feeding on some phytoplankton, but the direction of the arrow shows you that actually some of the zooplankton are eating the young herring too. As it gets bigger, it's feeding on the animal plankton, and that changes through its lifespan. So this is a hugely complicated picture, but it gives you some idea of why we need to understand plankton, the different types, and how they're changing. So we understand that we need to look at plankton, but how do we measure plankton? And if you get a group of plankton people in the room, as Katrina might testify from this week's meeting, we start arguing about the best way to do this, because there are as many ways to measure plankton as there are people who look at plankton. It's not easy, but basically most of the traditional methods come down to filtering it out of the ocean. So you get a concentrated sample that you can take back to the lab and look at with a microscope. And there are different nets that can do this. They're quite simple. Nets that you deploy off the back of the ship. This is the type that quite commonly used, I think, here in Prince William Sound. This is a more complicated system of opening and closing nets that gives you the ability to sample different depth layers through the ocean, but they're all basically the same, a filtering mesh that strains the plankton out of the water, enabling you to take it home. And I have a sample here you can look at later. This came from Prince William Sound, and this was quite deep, so it's about as big as plankton get. You look at it. More recently, we can use electronics to sample plankton. So over on the left here, this This is called a video plankton recorder, and it basically takes a photographic image of the ocean, and then you use the computer or your eyes to identify the different plankton from that image. Over here on the right is an acoustic assessment, so different frequencies of sound will give you an image of different sizes of plankton, and you look at this concentrated red spot, and that's where there's an abundance of plankton. You don't know exactly what it is, you have to get another method to know what type of plankton it is, but it is a way of assessing. With these two methods, you don't get an actual physical sample to bring home, you just get an image or data. But all of these devices, whether it be a net or a video recorder or a sonic system, they all need a ship to get you out to where you want to be to take your sample, and boats are expensive, especially when you're talking about being out in the open ocean. It can cost tens of thousands of dollars a day for a research ship to go out, so it's a lot of money to get an idea of what's going on. So the organisation I work for uses a slightly different approach, and it has a long, long history. And it started back in the 1920s, so we're going to step back in time now, with a guy called Alistair Hardy, who went on a sailing vessel. I can't imagine going all this way from the UK down to the Southern Ocean on a sailing ship like this, but when he was down here in Drake's Passage, he trialled his new invention to see how it would work. And he invented a machine that you could tow behind a ship that would sample the plankton. It was self-contained, it would do everything. It didn't need a person accompanying it. So it was robust. You didn't need to have a research ship that stopped to do a plankton sample and then had to start up again. So he trialled it. And his reasoning, this is where he was working back in the 1920s on the east coast of the UK. The North Sea was a very important herring fishery back then. This is the herring steam fleet all lined up outside the lab. And this is a photograph of the women who would follow along behind the herring fleet as it moved up and down the coast, packing the herring into barrels. And his idea was that if you could forecast where the herring food was going to be, you could send the ships there, and you would stop them having to search for the herring, and you could make it much more efficient. And I think what he got the idea from was that weather forecasting was taking off around this time. People were getting observations from lots of ships, and they were making forecasts of weather patterns, and people were using that. And that was his idea, so meteorology of the oceans. Let's try and predict where the fish are going to be. Now, other technologies came along and other understandings of how to find fish, so it wasn't used for this reason for very long, but nevertheless what he invented was a very successful instrument for tracking plankton. This is what it looks like today. It's not very pretty, it's not very glamorous, but it works. So it's robust and mechanical, and I've got a picture here of it sort of taken apart a little. So there's an outer towing body with a shock absorber and shackle that goes up to the ship. And inside it's a mechanism that's preloaded with filtering mesh and a system of wires that connect to gears. Each of these mechanisms lasts for 500 miles. So we send enough out to the ship to cover the length of tow that we want to do. And the only thing that the crew on the ship have to do is slide this one out and put the next one in, and it takes about 20 minutes to do that, and that's their only involvement. Everything else is self-contained. Inside this storage tank there's a preservative, so it fixes the plankton and keeps it in good condition until we get it back to the laboratory. So just a schematic of that. So the seawater and plankton enter through the front hole. They're filtered out by this piece of filter mesh here. There's a propeller on the back of the machine, so the act of towing this through the water behind the ship turns the propeller. There's a gearing system here which pulls the mesh through at a proportional rate. So if the ship slows down, it goes slower. If the ship speeds up, it goes faster. So you end up with a length of mesh that represents a distance of the tow. And the way I think of a sample that we collect is, think of a cylinder of water that's about the diameter of your finger, but 10 miles long. And it's the plankton from that cylinder of water that makes one sample. I've got some of the mesh we use here. So it goes through the machine very slowly. So this piece that I've got here is good for 120 miles. That's how long it will last. So when it comes back to the lab, we unroll it, we cut it into samples that represent 10 miles. And then we have to look at them and identify the plankton, and that's the time-consuming bit. So this is looking down the microscope at the mesh. The distance from here to here would be about 1.5 millimeters. So the really tiny little phytoplankton we see on the mesh. And then the bigger things, you can just see some of them here, we pick off and look at under a microscope. We identify the different types and we count them. We build up a database over time of where things are and how much there was. And these are some of the ships that we have that volunteer their time. So anything from a passenger ferry. This was an oil tanker that we used for a few years from Valdez down to California. This is a cargo ship that takes timber from Vancouver to Japan, brings cars back again. This is the container ship we use currently from Tacoma up to Anchorage, and other cargo ships. Even naval vessels have actually towed plankton recorders, although they weren't allowed to tell us where they were going, they were only allowed to tell us where they'd been afterwards. But fishing vessels, if they're on a long trip, have towed them. Anything really that doesn't stop and start. The advantage of using commercial ships is they leave port, they pretty much go where they're going as fast as they can. They don't stop and start, so you get a good, consistent sample that way. So the survey that I started working with in the North Atlantic started in the 1930s. This is their network of samples now over the decades. We started working in the Pacific in 2000. Southern Ocean has been sampled since 1991, and there's really recent surveys just starting around the southern tip of Africa. The east coast of the US has had a small survey since the 1960s, and Australia too for the last few years. So it's not a global survey, but it's not bad. We're covering a reasonable amount of the ocean. And each sample, like I said, is 10 miles long, so the Northern Hemisphere survey on its own has covered the distance of going to the moon and back 13 times now. So it's quite a lot of the ocean that we've sampled. We got involved in the Pacific because the Pacific is a huge ocean, and sending ships out into the offshore regions was just prohibitively expensive. So these red areas here show places where plankton have been looked at more than once a year historically. And there's not very many of them, they're pretty much restricted to the rim. So we started with 2 transects. Originally we went from California into Valdez. That only lasted for 3 years, and then we had to switch to a different transect from Tacoma up to Coconut. And we have one that goes across the Pacific 3 times a year too. And that's our survey coverage in the last 12 years. Just a few pros and cons, just to make the point. One of the advantages, it is cheap. We don't pay anything really for ship time. It's minimal freight to get the equipment there and pick it up. A bit of a thank you to the crew for their participation, but we're not spending thousands of dollars a day on a vessel. It's reliable. It's been run for so many years now. We know what we're doing, and it works well. It has limitations, but because we do the same thing each time, it's internally consistent and it tells us something about real changes. We can add other instrumentation and observations. I'll talk a little bit about that in a minute. And we build up this sample archive for future studies, and that's also an important thing I'll talk about later. But there are some disadvantages. Analysing all those samples is labour intensive, and that is expensive because that takes people sitting down at a microscope. The method has limitations. It samples at a fixed depth behind the ship, and plankton are 4-dimensional. They change through the 3 dimensions of space as well as through time, and we're not measuring all of that, not able to capture all of that. Because we're using commercial ships, we don't have any control over where and when they go. We get an idea of their schedule, but they're not going to change what they do for us. The commercial reasons behind what they're doing are most important. So, and although they're all very willing to help and get great satisfaction out of doing it, we are still low on their priority list. We're not going to jump ahead of getting into port at a certain time. And sometimes we don't get exactly the information we need. There's been a few times where somebody pulled the CPR in but forgot to tell the bridge, so when they eventually remember, they're not exactly sure when they pulled it in, and that makes it hard for us to reconstruct the tow. So sometimes there's a few issues with the information we get back. and give you one of the examples of the issues with a ship of opportunity survey. On the left here I've got the transect from Tacoma up to Anchorage. Now there's 53 separate transects there, but they overlay each other almost completely every time. That ship makes the same trip. This one was actually a little tow we did on its return from Kodiak. When it leaves Anchorage, it goes down to Kodiak and then across to Tacoma, so this is a different one. And we just did a little bit of the return leg to fill in a gap one year. But otherwise, it's pretty consistent. Over here, this is the ship that leaves Vancouver and heads over to Japan, and there's only 33 transects here, but you can see they go all over the place. They pretty much all go through Unimac Pass, but to get there, they take a different route. Mostly that's because this is a longer transect, it's a slower ship, and it makes economic sense to avoid storms, so it will go out of its way to avoid the storms. So if you're looking to build up a time series and to be able to see if something's changed, this is really good because you know you're sampling the same area every time. On the other hand, if you're trying to build up a big picture of how things are over the whole ocean, maybe this is a bit more useful because you're covering more of the ocean as long as you keep going long enough. So a few examples of what we found out. There are many examples I could give. A lot of the work is focused on looking for changes due to perhaps climate change, effects of overfishing, particularly important in the North Sea, which is a very heavily fished area. Just the natural variability in the plankton, and impacts of one-off events such as oil spills or volcanic eruptions that have caused changes too. So all of these things have been looked at. I've got a couple of examples here. This is a plot, just to orientate you, this is the UK, and this is the coast of France and Spain. And we've got time periods here moving from left to right, so back in the 1950s to 1981, 80s to 90s, early part 2000s and the middle part of the 2000s. Up here is a group of warm water plankton, and down here a group of cold water plankton. And the maps show the redder it is, the higher the numbers. So back about 30 to 50 years ago, the warm water plankton were concentrated down off France and Spain. Gradually, as the waters have warmed, and they haven't warmed very much, but it's just enough to enable the plankton to move up. They're usually following the shelf edge current, but the waters up here now, right around the top of the UK and off of Norway, are now sufficiently warm that the warm water plankton are quite well established and doing quite well. At the same time, the cold water plankton are kind of moving north. We don't sample up into the Arctic very often, but you can see that the red has retracted, and it's a much smaller area now in the Atlantic. So why does this matter? Well, species like cod feed on a particular type of plankton. If their preferred prey is moving away, they will probably have to move to follow it. This is another example that we think is linked to global change. So this is a small diatom. It's very common across the North Pacific, and we first did our first pilot sampling in 1997 in the Pacific. And in 1997 we noticed up in our samples in the North West Atlantic that we found a few of these. We thought, oh, that's odd. Or maybe it's contamination, because now we've got some Pacific samples. But we found it every year since, and other people have found it too. So what we think is happening is that as the Arctic ice has been declining over time, movement of water from the Pacific to the Arctic has increased through more of the year. And it has enabled this particular organism to come through and set up residence. Now, this used to be common in the North Atlantic, but that was 800,000 years ago was the last time it was recorded there, so from sediment samples. So it's not like it's a harmful species, but probably, if one thing is making this trip, probably other things are. Maybe bacteria, maybe viruses. Maybe things are going to be coming the other way too. So something that a monitoring program is very useful for is looking for those kind of changes that may happen. And just, this is my area that I've been looking at in the Pacific. So this little box here around Cook Inlet and the shelf just to the southeast of it. And these are just time series that we've got for the last 12, 13 years. And on this upper one, it's called an anomaly plot. So all you need to know really is that above the line, numbers were higher than normal. Below the line, they were less than normal. And this is the temperature, just the mean temperature, the average temperature for the year. And in years where the temperature has been warmer, in the first part of our time series, we had generally higher numbers of the plant plankton. And then as temperatures have gotten cooler, we find lower numbers. And the zooplankton, as you would expect, have followed that. So when their food source is higher in number, they've been higher in number too. And in the last few years, we've seen some lower anomalies. So again, we're monitoring what's going on. At the moment, this is just climate variability. We've had a cold period in the last few years. That may change. We may see things go the other way. It may last for a bit longer. One thing we did for a 5-year period was we had an observer on the ship that goes from Vancouver to Japan, and he sat up here. This is him in the snow, probably somewhere in the Aleutians, and he was recording seabirds and mammal observations on that trip. The very first time I sent him out, well, I didn't send him, but we asked if he would go and do it. It was the Stanley Cup finals, and his team were in the finals, and he wasn't very happy about going. And it's just like, that ship is leaving then, sorry. When he got back, he said that was one of the most amazing things he'd ever done, and he would miss his own wedding to do it again. So I'm not sure how I felt about that. So, I mean, just going through Unimac Pass, there were just millions of seabirds. It's a very productive area. So he did that for 5 years. He did 16 trips. He wouldn't let anyone else do it. So, and then our money ran out, unfortunately. But the reason I put this up is just to show, this is just some of the seabird data they collected. The bigger the spot, the more abundant. These are some of the different species that they recorded. But again, well, it's not the best platform for recording birds and mammals. It's a long way up from the ocean, and the ships are moving fast. It was the only way to really fill in some of these remote areas where people had very few observations. And it's something we're hoping to do on our transect into Tacoma. There's quite a lot of interest in having an observer on this transect going up here. So I'm hoping in the next year or two we'll be able to get that started, because it's really nice to be able to tie in seabirds with some of their food sources too. And just a couple of little comments about things we might do in the future. And I say future, but we've actually started these in some ways. There's just things that need some further development. So there's been a lot of talk lately about plastics in the marine environment, the big plastics that are floating around causing problems. But as these plastics degrade, and in fact small plastic fibres, so this is a particle here that's only about 1/4 millimetre across. But we find them in our samples, they're prevalent. One advantage of having an archive of samples that goes back decades is we can go back and look at those samples and see, okay, when did we start catching these, and have they spread over time? So we have a student at the moment who's just started looking at that. This isn't the easiest slide to read, but the dark colours show where on the samples that he looked at, there were something like 8 to 25% of the samples had microplastics on them. The pale colours, there was somewhere between 0 and 2%, and in here it's 2 to 8%. So this is over the North Atlantic, but there wasn't really anywhere that didn't have plastics, and this was just a 7-year period we looked at from 2004. So we're hoping that we can go back in time and look at some of the earlier samples from the 1950s and start to see when and where these plastics started appearing. And I don't profess to understand this at all, but this is using the DNA techniques to look at DNA on the samples. So all I know is this particular little species here, it's really, really hard to see even with a good microscope. And even if you do see it, you can only identify it as one of 2 types. In actual fact, there are more than 30 different species, and 12 of those species form the harmful algal blooms that cause problems with shellfish. And if shellfish eat the plankton, they concentrate the toxins, and if we eat the shellfish, we get sick. So it's important to know if they're around. And we can't do that with a light microscope, but one of the women in our group has been looking, and these are all the different types that she found. The ones not coloured are the reference types, and the ones coloured in blues and greens were from the oceanic samples, and the orangey ones are from the shelf samples. So, and she found a new occurrence of one of the species too. So again, these kind of techniques are quite expensive still at the moment, but they will get cheaper, and they will enable us to look at things we can't see with our eyes or with a microscope, and bacteria, viruses, small things. And that's another way of having a sample archive. You can go back and look at some of these things. When did they start appearing, and how common are they? So I'd just like to end with a big thank you. I went to visit the ship when it was in Anchorage earlier this year, and the captain and crew were very, very hospitable. So thank you to the Horizon Kodiak, which has been a really great supporter of our program now for almost 10 years. We have a lab in Anchorage that help us out by picking up the device and servicing it for us in between times. There are quite a few people who spend a lot of time at the microscope to get these data, and it's a very valuable contribution. And of course, the people that fund this, the XMLD's Ausable Trustee Council, North Pacific Research Board from this part of Alaska, are very big supporters, as well as Fisheries and Oceans Canada. Thank you for your attention. I left my email address up there if anyone wants to follow up, but I'm happy to answer any questions now too.
Sonia, since I know some of the internal stories, it's not fun to talk about pitfalls, but when you talk about reliability, the machine is often more reliable than conditions it might encounter in the oceans that are inhospitable. So I don't know if you want to tell folks about some of the big challenges that you encountered and like what that cost is to the program.
Anyone who works on the ocean knows that it's a hard place to work. Anytime you put something in the ocean, you don't know for sure if you're going to get it back. And that goes for fishing as well as ocean science. So we've been pretty lucky because it is a robust machine. The ships don't slow down to deploy it. Some of them are going over 20 knots and they just throw it off the back, and they're pretty robust. And when they come back in, they often clang against the side of the ship. We did lose one last year. We're not sure. When the crew recovered the tow wire, it was sheared off really cleanly. We think that it hit something like a submerged shipping container, because some of them float, maybe 5-10 meters down, and the ship can go over the top, but the CPR just went straight into it. And we suspect that maybe something to do with the tsunami and the Japanese debris that had come across. So that happens occasionally, and of course then you've lost the device and you've lost the samples, and it's one of those costs you just kind of have to take a deep breath and suck it up. It's more rugged than some, at least, and because there's no electronics involved, it doesn't matter if you throw it off a fast ship, it's still going to work. So most of the time, as long as it doesn't get lost off the ship, we get samples back at least 90% of the time. So it's pretty good.
Have you had a chance to look at the plankton data overlapped onto the bird data, and does it correlate pretty well?
Not as well as we expected. Yeah. When you sort of like group everything together and say, is the total amount of zooplankton related to the total amount of birds? It doesn't look like that. When you start teasing apart the different types of plankton with different types of seabird, there were actually some reasonably strong associations. But there's a lot more to the story than just saying there's this much plankton, there should be this much birds. So there's a lot more.
But if you had lots of birds, you have lots of plankton going that direction?
Well, sometimes you have not much plankton because you've had a lot of fish.
A lot of fish eating and the birds
eating
fish.
Fish.
Yeah,
so, it's one of those things
what
that,
you got there, right?
Yeah, we don't have the fish story in the middle because that's even harder to do from a ship like this. But you hope that if you come at it from two ends, you might be able to say something at least.
Sonia, did you say what the mesh size is that you're using?
270 microns.
Okay, so is it going to get pteropods?
Yeah, we catch pteropods. Yeah, there's quite a few different types. They do get a bit crushed by the mechanism. That's the other thing, that when you, because these things basically end up as a flat sandwich. We call it roadkill in the plankton world, because the pictures I showed are very nice, plump, juicy, round plankton, and when we see it, it's not like that at all. So, but yeah, this is actually real silk. It comes from China, and it was originally designed for sifting flour. But being silk, it's quite thin, and as a natural fibre, it's actually quite sticky. So it collects quite a lot of plankton that man-made fibres don't do quite so well at, which is good. But yeah, 270 microns is the mesh size, so about 1/4 of a millimetre, which is in the range of what most plankton samplers use.
And does that take the sample and then fold in on itself when it gets rolled up? Or
there's two layers.
2 layers?
There's 2 layers. So you end up with plankton that filtered by this piece then comes over the top as it rolls on, and you get basically a long roll of the sandwich, and they're trapped between the 2 layers.
I don't know if you enjoyed it, but I liked the analogy that our own biological oceanographer Rob Campbell made this week, which is, it's basically like when you take a long car trip, a road trip, and all of the insects get smashed on your windshield, and then you do a scientific assessment of distribution and abundance and all that.
Yeah.
So you did show abundance, changing abundance of cold and warm water species in the North Atlantic. Do you all anticipate that anyone will look at things like, are you able to look at condition of plankton, such as how they might be affected by ocean acidification in the North Pacific, which is thought to be a bigger issue here maybe than in the Atlantic?
Yeah, we've looked at the Atlantic samples, and at the moment, the temperature is a more important effect. The temperature is actually causing an increase in some of the organisms there, rather than ocean acidification causing a decrease. I think there will be something we can do looking at shell thicknesses and things like that, but we don't have as long a record here yet as they do in the Atlantic, so they, in some ways, although it's not such a vulnerable area there, they stand a better chance of seeing a change over time because they may have a 50-year time series to go back to. But there's certainly a lot of interest in looking at those kind of organisms that are particularly sensitive.
How many people at Sappos are involved in CPR studies overall worldwide at your institution?
Everyone in Sappos is involved in CPR studies because that's pretty much all we do. There are probably about 15 people who spend time at the microscopes, although a lot of them are part-time, and then there are researchers on top of that. But some of the surveys around the world are completely separate. I'm kind of unique in being out here in a different part of the world, but still working with them, whereas some of the people, some of the surveys are completely standalone and have done their own thing for many years. We all work together, you know, and we sort of compare notes, and we've just sort of got a group together to do some inter-comparisons and pull our data together. But we are sort of separate groups mostly.
Are copepods increasing or decreasing in abundance?
Depends which type.
Yeah.
One of the things I didn't show, and they're not a huge contributor to the community, but when we've had some warm years, the warm water copepods have come up further north in the Gulf of Alaska. Now they're smaller and they don't have as much lipid in them. So if they were to become more abundant, you know, if the ocean warmed up and they became more established and more abundant, that does have a consequence for the, you know, for the animals that feed on them. Because the typical copepods we have here are actually really big and juicy. People back in the Atlantic are quite jealous because we have some of the biggest, chunkiest copepods out here. A lot easier to see. You can sort of look at them with the naked eye and say, oh yeah, that's one of those.
Great, thank you very much, Sonia.
Thanks for coming out on Friday night.